Well, thank you. So this is visible to everyone, I hope. So thank you so much for inviting me. And I feel sorry, but also mainly sorry for myself that my trip got derailed yesterday. United Kingdom, as you might know, is a very underdeveloped country where a power surge can shut down a terminal at the third largest airport. So I would have missed my flight, and it was very difficult to organize to come on for a 2-day visit, sadly. But also it's good that we have internet. Yeah, so thank you all for coming. So what I want to tell you a little bit about is the work that I've been doing, and also what a lot of people in my lab group now have been working on since I started a group in Manchester. And that's really this broad idea, broadly speaking, the idea of how we can provide a mechanistic understanding of gene regulatory evolution in order to better understand the basic fundamental principles that components that come and shape how systems and organisms evolve. So very broadly speaking, why do we why do we even care about promoters? And I'm focusing this talk on promoters as one of the key aspects of gene regulation in prokaryotes and bacteria. And we care about promoters ever since the early days of these two compatriots of several of you, Jacob and Monod, who discovered the lactose operon. And they generally discovered that the binding of proteins to DNA is a very key and fundamental process in biology, so much so that later on it became more and more evident that that process of gene regulation and regulating when each gene in the cell is produced and how much of it is made really lies at the heart of biological diversity that we see. The extent to which is arguable, but it's certainly a key component that's led to diversification of life. So it's an important phenomenon to look at. And so these gene regulatory networks that consist in bacteria of promoters and proteins called transcription factors that bind to them, they organize, as I said, into these large-scale networks. And the reason why I'm putting this here is really to illustrate a general point that particularly when evolutionary biologists looked at gene regulation, vast majority of those efforts have focused on understanding how these big networks. By the way, can you see the mouse that I'm moving? Is this visible?
Yes.
Yeah.
Yes, it is.
Thank you. So evolutionary biologists particularly have focused on how these large structures change in time. And also from a more, let's say, mechanistic standpoint, how the starting structure of the network affects the way it can change under some kind of evolutionary pressure. So in a sense, one can think of vast majority of work on gene regulation as being top-down, understanding what happens at this very large scale level, as opposed to bottom-up, which would be to understand what happens when you have a single, what happens at the level of the single node, the single protein binding a single promoter, what happens there, and are the laws that govern how that process works may be involved in shaping this overall network. So that's in some ways kind of a vision for my group, but it sets out the general idea that what we're interested in is the relationship between mechanisms and molecular mechanisms and what they mean for evolution. So let me first define what I mean by promoter, because particularly if you work on eukaryotes, this image gets a lot more complicated than what I'm about to present. So promoter in bacteria, what I'm going to call a promoter is if we do have a gene of some sort, doesn't matter what it is, it's the DNA sequence before that gene. This is very much not to scale, by the way. It's a sequence before that gene, which determines through the binding of one or more molecules how much of this gene you're going to make. And so in the simplest possible case, so the minimum requirement for a sequence to act as a promoter is that it binds RNA polymerase. RNA polymerase connected to one of several sigma factors, but nevertheless, RNA polymerase you need in bacteria because that's what initiates the process of transcribing a gene from DNA into RNA. And when the promoter binds only RNA polymerase, we call it constitutive. However, when it binds one or more transcription factors, we call it regulated. And those transcription factors can be repressors or activators, but actually I'm not going to talk about that. Today I'm going to focus only on constitutive promoters. And the reason for this is that we specifically wanted to start with simplest systems one can imagine, in order to really try to nail down what are the mechanisms of their function and what do those mechanisms mean for how the system can evolve. So today I'm going to tell you really just the simplest possible story, which we are extending slowly in various ways in my group right now. But this is the starting point, I think. And so we also don't focus on any kind of promoter, but rather specifically on promoters that are regulated by sigma 70. And we study this in Ecologie. So sigma 70 is the most common sigma factor, which brings RNA polymerase to regulate expression of about 70% of all the genes in Ecologie. And so thinking of how looking at promoters, how do people generally study promoters? Well, on one hand, there is an attempt to understand where the promoters came from, just like with any kind of sequence, where does it come from? Another option is to study how the promoters work mechanistically. And the third, maybe more recent option is to really just care about using them. And I outlined this in this very sort of almost cartoonish way because I want to align these ideas with the general fields of evolutionary biology, molecular biology, and synthetic biology, all of which are interested in different ways in promoters. But what I think is really quite important to outline is that the way in which the way in which promoters, just like any other system, is studied by these different fields is very different and often not very interactive or cross-disciplinary. So in a sense, when you're a molecular biologist, you study really the mechanisms, how it works, without caring what that means for evolution. And while I understand that there is such a field as molecular evolution, which is a very large area, the focus of that field is largely on identifying which mutations at the DNA level are responsible for adaptation, which is not really what a molecular biologist does. They're interested in how things work. And so that's, from another perspective, the real motivation for me, and I try to inspire that same motivation in the people in my group, which is really to try to connect the fields of molecular and evolutionary biology. And the field of synthetic biology is actually very useful and provides a lot of tools and techniques for doing so. And what I think is the really important thing to emphasize about creating such a connection is that by doing so, by understanding how the existing mechanisms in the cell shape the way in which an organism can evolve, you start being able to generalize beyond the specific system you study. So you can take the system you study and understand the principles, the mechanisms that are then maybe related to other systems of interest. And this allows you to then predict evolution, hopefully in a more comprehensive way, and in a way that can be extrapolated beyond the specific system that you spend a lot of time and effort experimentally understanding. Or in other words, the way in which one can see what this philosophical rambling that I'm telling you might mean is that we're trying to make the process of predicting what the mutations do and how they interact with each other better and more predictable across a range of systems. And this is a fundamental, what mutations do is a fundamental property of evolution, and the one that is proven to be one of the most tricky properties to actually predict for any given system. So how do we study promoters? How does this actually play out, this philosophical rambling that I just told you? What does this actually mean? Well, it means that first we experimentally probe synthetic promoters. And the reason why I say synthetic is because we want to make things that are controlled in as many aspects as possible so that we can actually understand the relationship between individual mechanisms of function and what happens without all the other biological complexity that comes into play. We also, then we use that data to understand the mechanisms that govern how, in this case, promoters function. And then we develop models, ideally predictive, and certainly with an emphasis on mechanistic models. And then we want to use those models to say something about evolution of similar systems that go beyond the specific experimental system that we focused on. And in this talk, I want to give you two examples of how we did this in the past and what they meant, what they allowed us to do and to say about some evolutionary properties of promoters. So first I want to outline something that I'm going to refer to in this talk as a standard view of bacterial promoters. And the standard view of promoters is that, again, definitely not to scale. When you have a gene, another thing you need is a ribosomal binding site, which I'm written here as RBS. And then you have the promoter. And the traditional standard view of promoters is that there is one binding site for RNA polymerase, typically identifiable by these -10 and -35 boxes that have a specific clear sequence that they like the most. And if you look at, for example, bioinformatic approaches to try to predict where promoters are in cells, you find that they really follow this assumption very strongly. There is one site and they just try to align this ideal binding site. Right, so one binding site for RNA polymerase to DNA is the standard view. And for the first part of the talk, I'm going to tell you about how we use this view of promoter to say something about epistasis between mutations in the promoter. So the first thing we do, as I said, is we experimentally probe the synthetic promoter. So what we're looking at, as I said, is we're looking at the interactions between mutations, so epistasis. And because epistasis is such a complex term, I'm going to actually define it here specifically so we're all on the same page, because there are a lot of combating definitions or confusing definitions. However, the reason why we're focusing on epistasis, as I think to this crowd will be extremely obvious, is that it's what shapes the fitness landscape of an organism, turning it from a single peak when there is no epistasis or some type of epistasis, and having the potential to convert it into a multi-peaked fitness landscape, where as a result of having a specific type of epistatic interactions between mutations. So back to the definition of epistasis. So if we take some quantitative phenotype, and because we work on gene expression, that's what I've written here. We start with some wild type, which has some fictional gene expression level. Then we can think of introducing two single point mutations individually, either a mutation A or a mutation B into the wild type. And in this illustration, both of these mutations have a positive effect on expression level, so they both increase expression level. So then the question is, what is the double mutant? And we can adopt either an additive or a multiplicative expectation. Let's stick to additive here, just for the sake of illustration. So according to an additive null model, if these two mutations do not interact, then our double mutant would be the sum of their effects. So this effect A, so this effect A plus this effect B together should give us the case of no epistasis. If we then experimentally measure our double mutant and find that it has higher expression level than what we predict based on individual mutation effects, we can say that this system is in positive magnitude epistasis, and conversely, it can be negative magnitude epistasis. It can also be in other types of epistasis, but I'm not actually going to talk about those because that picture gets a little murkier. Well, not murkier, just not applicable to the promoters, at least at the level that I'm talking about. So now that that's the definition of epistasis, how do we study it? Well, let's take our experimental system, which in this case is a simple yellow fluorescence protein marker gene under the control of our promoter. And we start with a very strong promoter sequence, which is derived from the lambda PR promoter. It's a very well-studied system. So we start with a very strong promoter and put it into a plasmid system in E. coli that we study. And then we introduce mutations into the promoter. And in this study, we created 240 random double mutants together with all the corresponding single mutations so that we can calculate the prediction for epistasis and compare it to the experimental observation. And so when we do that, we can measure the effects of all of those double and single mutants and calculate epistasis along the idea that I told you before. And then we can get a certain representation. We can show you what that epistasis looks like for these 40 random double mutants. And what you're looking at here is the additive prediction, so the null model for the interaction based on the single mutant effects, and then the actual experimental observation. So the deviations above this line, so the mutants that are above this line are in positive epistasis, and the one below in the blue area in negative epistasis. And to a large extent, I think this part should feel very familiar because this is really how vast majority of studies of epistasis work. We take a system, we perturb it, and we measure those observations. Where I think things get maybe a little different, it started for us with a simple kind of conversion of this plot that's in front of you into a slightly different format, which is simply showing, here I'm showing you just the number of double mutants that are in positive and negative epistasis, with the darker parts of the bars representing those that are significantly different from this no epistasis line. And what we see here is somehow was motivating, although in retrospect, I'm surprised that it did motivate me to look into this further. But there was a pretty evident bias. There is just a lot more double mutants that are negative than those in positive epistasis. And it wasn't immediately obvious to me or people I was working with why that would be the case. So we tried to see how we can explain why there's so many more double mutants in negative epistasis. And to do this, we turned towards a mechanistic model, which is a very simple model based on the thermodynamics of binding between DNA and a protein. And this model, in fact, is so simple that it really considers only one of many aspects of how gene expression levels are controlled. And that is simply the energy of binding between a protein and the DNA. Right? So that's the only thing that we're actually looking at here. That is a process, however, that lies at the heart of regulating gene expression in promoters, but it is also a very simple model. And in this model, when you look at the energy of binding, so when you consider the energy of binding between a protein and a given DNA sequence, so the given promoter variant, you can translate that by, you can envision and calculate the likelihood of promoter finding itself in one of the two states. One state being the off state when the RNA polymerase is not bound, and the second state being the on state where RNA polymerase is bound. And we considered so we can, based on the energies, we can calculate the probability of the system being in this state. Sorry for stuttering there a little bit. And then we just simply say the probability is related monotonically to gene expression level. And what's really key here, that should be obvious to those of you who have even who have some understanding of thermodynamics, is that the relationship between this energy of binding and the probability of being in the bound state, which is what we call gene expression here, is not a linear relationship. And also note that this is always a little bit confusing, at least for me. Lower energy of binding means stronger binding. So the lower energy means you bind more. That's why this looks inverse. And so what's a really key point here is that the relationship, due to Boltzmann equations, the relationship between the free energy of binding and the likelihood of being bound is not linear, but rather it's a sigmoid, which actually can be, for a vast majority of the relevant energy range that is measurable in the lab, can be portrayed as a function, which I'm confused with. I'm blanking out on the name, but this kind of function. Sorry about that. And so why is this important? Well, it's important because it lends itself to some pretty cool predictions about epistasis. And to illustrate that, let's start with some wild-type sequence. Just imagine some wild-type sequence has certain energy of binding leading to certain, in this case, very high expression level. Then we can introduce single mutations into the system, either mutation A or the mutation B. And each of these mutations in this example, what it does is it increases the energy of binding, which means it decreases expression levels. So what is the prediction? What is our null model for epistasis? Well, the null model for epistasis here is the additive one. So the double mutant should be somewhere along this line. But of course, it cannot be on that line, because I just told you that all the mutants, every possible configuration of energy to gene expression has to be on the dotted line. So what we end up here is, in this case, we end up with the double mutant that's in negative epistasis, right? Simply because adding these two will always be on a line that's curved in that way. It will always be below. And in fact, what's really fascinating here is that we can actually predict the sign of epistasis based only on the sign of individual mutation effects. So all we need to know is whether the individual mutation is going to have a positive or negative effect, and same for the second mutation. And based on that, in promoters, you will always get this very easy relationship between the sign of individual mutations and the sign of epistasis. And I think that's really cool, because epistasis is thing that's very difficult, a phenomenon that's very difficult to predict. And here we can actually predict the sign of epistasis based only on individual mutation effects. And this actually explains this observation that started our interest, because remember I mentioned to you that our starting promoter is a very strong one. So this wild type is always on this high position. So vast majority of mutations that were introduced actually are in this negative scenario, which means that we have negative epistasis, and that's sort of what we observe. We have in this paper a much better and independent verification that our predictions are accurate, but I'm not going to show them to you now, because really what I wanted to outline is that in this case, adopting a mechanistic perspective for what mutations do actually shows that even the very notion of epistasis, the way we define it as an additive model, biologically doesn't really make any sense. It makes sense evolutionarily, and that's an interesting, that's an interesting problem in a way that I don't really know how to reconcile. But the point I wanted to make here is that knowing the biophysics and how this mechanism works, the idea that epistasis should be here, it's actually biologically nonsense. And so I think that leads to a pretty cool conclusion that we can actually predict epistasis, or at least some aspects of epistasis, without really having to do that many experiments, just knowing what a single mutation can do. That's great, but that's a very, very simple model. And there are many pretty obvious problems with this model, but I'll tell you one that maybe is not that obvious, but is actually really important. And that is that when you use this kind of modeling, you typically try to model mutations around the given promoter, and that given promoter is what you develop your model parameters, what you derive your model parameters from, right? And when you do that, you can get pretty high predictability. So this is an R-squared fit between model predictions and experimental observations. What you get is pretty high predictability of the local data set. So data set around the promoter that you mutated originally, that you used to get your model parameters. However, when you apply this to random data sets, to sequences that are far removed from your starting position, the model actually starts collapsing quite rapidly after 3 or 4 or 5 mutations. It just doesn't work as well. And that's a problem, right? Because it's a problem which tells us there is something else going on here at the mechanistic level. So it's not just that binding in that simple way, conforming to that standard view of the promoter, that explains the variability, the sequence to the genotype to phenotype mapping for at least constitutive promoters in bacteria. So this was the second challenge that we posed ourselves, and I'm going to tell you this. The next part of the talk is about a paper that we are currently in the process of responding to reviewer comments, but hopefully there is some version of it on bioRxiv which we haven't actually updated in over a year, so it's not quite the version that we have right now. But the question we ask ourselves is that standard view of promoters which I told to you about before, is this actually realistic? Is this a realistic view of how promoters work? And why might it not be realistic? I should say, we follow the same kind of procedure here. We have some experimental probing of synthetic promoters that helps us understand the mechanisms, that helps us develop the model, that then we use to explore some consequences of that model for evolution. So again, very same system as before, a promoter, some promoter sequence that regulates the expression of a yellow fluorescence marker in a plasmid in E. coli. These are, by the way, low copy number plasmids. They have only 2 to 3 copies. Really unpleasant to work with, but very useful because they look like chromosomes in most ways. So we probe the system, except in this case, we do it randomly. So we randomly mutate the promoter, and I'll tell you 2 different ways in which we randomly mutate it. And then we do what's called a sort-seek experiment. We split all our mutants based on their fluorescence, based on their expression level, into 4 bins: no, low, intermediate, and high expression levels. Then we sort that, we sort all of those mutants, and then we PCR tag them. And this allows us, when we sequence them in mass, this allows us to kind of reconstruct the estimate of expression level for each mutant based on which bins and in what quantity the copies of that mutant were found in our sequencing results. That's a sort-seek experiment. And based on this, we have a map, we get mapping between specific mutants and their expression levels for a much larger library than 240 mutants. In this case, tens or even a couple 100,000 mutants in here, in some of these libraries. And we do this for two different libraries, which I'm here portraying on this fully realistic, since I'm so interested in realistic landscapes, I like to portray them as two-dimensional. This is the entire genotypic space, which is completely two-dimensional. We created two experimental data sets here. One is around the lambda PR promoter again. This is the same promoter we used before, with 12% mutation rate per position. So each position for each mutant has a 12% chance of being mutated, which means 4% to be any of the other three positions rather than the wild type. Bases, not positions, sorry. The second library we create is called the 36N library because it's 36 completely random base pairs in the middle of that promoter. And they're completely random with 25% chance of being any of the four sugars. And the reason why we do this is because that provides us with a local data set. And we use this local data set to derive model parameters, to train the model, and then we separate 60% of it to validate the model. But then we have the other global library, which the model never sees. And this is a really key feature of what we did, which is what makes us trust us, what I'm going to tell you a lot more, which is that we never trained the data on this model. We simply use it to validate the model trained on the local library. And so what are the things that we introduced to this thermodynamic framework that I described to you before that might be different, that might capture some of the complexity of how models work, or how promoters work? Well, there's a bunch of mechanisms that we use. One is actually a well-known aspect of how promoters function, which is that the spacer, the distance between this -10 and -35 foot is variable. Typically, it's 17 base pairs, but it can vary plus or minus 2, typically from 15 to 19 base pairs. Now, that's known, but what we do in our model, we actually calculated the energy cost for RNA polymerase changing its conformation to go from 17 to 16 or 15 base pairs. The other mechanism of promoter function that we included here is really we allowed RNA polymerase, we didn't think that it has to bind to one specific location, because as long as RNA polymerase binds anywhere so that it transcribes this ribosomal binding site in the gene, that's a viable transcript that's going to result in us measuring some fluorescence. So it doesn't matter if RNA polymerase binds here or maybe a little bit upstream or a little bit downstream. In fact, all of those positions would result in a viable transcript. So we allow polymerase to bind in multiple locations. That also means that some of those binding locations might be unproductive. So imagining that RNA polymerase now binds here, so the transcriptional start site, let's say it starts in the middle of the ribosomal binding site. Well, without the transcribed ribosomal binding site, you're not going to have any protein. So that's a non-productive transcript. Similarly, RNA polymerase can bind on the opposite strand, and that can interfere with our promoter function. So that's also unproductive binding, what we call. We also look at the interactions between positions in the RNA polymerase binding. So this is different from the epistasis I described before, because the epistasis I described before is sort of almost like tricking, telling you to do wrong maths to tell me that biophysics looks, doesn't look the way it does. What we measure here is actual epistasis at the level of energy. So not at the level of probability of binding or expression, but the level of energy. And that's in some sense more reasonable and meaningful way of thinking, or at least biologically meaningful way of thinking about epistasis, not necessarily evolutionarily, which is also an interesting thing. And finally, we look at the model how quickly once bound RNA polymerase runs away from here and actually opens up the DNA and makes mRNA. So just to illustrate what I mean by this in a slightly different format, if you look at here, this is the same plot I showed you before. This is maps to the specific RNA polymerase binding site, and this is just an illustration of what the transcript looks like. In our extended or full model, as I call it here, you can see that we allow different spacer variations, which you can see here. 0 corresponds to 17, and these are the variations of it. It also allows different binding configurations along the promoter, some here shown in blue being productive and the red ones being unproductive. So we evaluate all of these new mechanisms using the PR library, so the local library. And what we find first is that all of these mechanisms are actually important. They significantly improve the predictability of our model. However, some are more important than the others, particularly spacer. Knowing the energy of varying the spacer is very key, and allowing for multiple binding is really important, particularly at low expression levels, which is interesting in its own right, but I will not talk about it. So this is where we end up, right? This is what I showed you before. This is the standard model, which we also developed it and trained it on the same library, just without all these extensions that I told you about. And it's quite good and very good, I would say, at predicting the variability within its local data set, and pretty not good when predicting the random library. So our extended improved model, it improves the predictability of the local data set. This looks small, but it's actually, given the size of the data set, extremely significant, but it's not an impressive improvement. However, the real impressive improvement, to me at least, comes when we apply this model to random data set. And what this is indicating is that simply knowing and incorporating this new, pretty basic mechanisms of how promoters work, we can actually overcome one of the big flaws of this model, which is that it's just not very good at predicting expression levels from random sequences. And we validated this in a variety of ways using RNA-seq data, using other people's data, but I'm not going to show you any of that. For that, you'll have to wait for our paper to one day be published when all the collaborators decide to do what they need to. Ignore that. Yeah, so the really key point is that the inclusion of these mechanisms gives us confidence that I think we can, with reasonable confidence, now say that we can predict the constitutive expression levels from random sequences in E. coli. What's really cool and interesting about it is that then it allows us to ask a bunch of evolutionary questions. So one simple evolutionary question we can ask is, what is the likelihood of a random sequence being a promoter? Now, of course, that depends on the size of your sequence. In this case, we model 115 base pairs, which is the average size of a promoter region in E. coli. And what we find is that on that sequence, something like between 15 and 20% of all random sequences are predicted by our improved model to lead to transcription, to some level of measurable transcription. So this is something we measure in the lab, which we know a lot of stuff happens below that threshold that's relevant, but this is already above a pretty high threshold for biology, will lead to some, so about 15 to 20% of sequences will lead to some expression, completely random sequences. What's even more is that we can take those sequences and look at what happens when we find one mutation that has the biggest effect on their expression level. So here, along the x-axis, you have the predicted expression level, which is the log peon, predicted expression level of the original random sequence. And what you find with this line, on the right of this line, are sequences that have expression, on the left, the ones that don't. So this is our 15 to 20% that has expression. Now we look at one single mutation in each of those random sequences, and we simulate this for 1 million sequences, I believe. So for each of those million sequences, we find one point mutation that has the biggest effect on changing the energy of binding of RNA polymerase. So this is now the predicted expression level of that strongest effect mutation. And what we find is that mutations that go from the left into the above here, so this gray area, are the mutations that go from non-expressing to expressing due to the single point mutation. And this is actually almost 90% of all random non-expressing promoters. You can find at least one point mutation that will take you from no expression to expression, which I think is pretty crazy. And when we sum this up across all possible random mutations you can have, single point mutations you can have in each of these randomly generated promoters, we find that something between 1 or 2% of all random mutations in a promoter lead to expression. And I think that's pretty fascinating because so much of thinking in evolutionary biology is how do you evolve a function? And really, when it comes to promoters, oh, sorry, actually, I shouldn't have come to that yet. I'll come to that conclusion in a second. I forgot that I want to tell you something else that's interesting, which is that this ease of evolving promoters is actually reflected in the E. coli genome. And one thing that has been shown before prior to our work is that within coding regions, you have very strong selection against promoter sites, binding sites for RNA polymerase. So this is in this green-blue color is the actual number of promoters based on their strength, based on their free binding energy. So there's a binding sites, RNA polymerase binding sites based on energy and their number. And this is the number predicted by scrambling the genome. And so you can see that there's a big difference. Now, what's really fascinating is that we see the similar evidence of selection against RNA polymerase binding sites, even when we look at only known promoter regions. So this is from RegulonDB, which is a database that has experimentally verified regions that act as promoters. And even in those regions, we find evidence that there is selection against having too many sigma 70 RNA polymerase binding sites. And this is now back to the conclusion I wanted to make earlier, which is that contrary to most search of evolutionary biology for how new function emerges, with promoters at least with promoters in bacteria and constitutive promoters in bacteria, the real question is not how these promoters evolve, but rather how do they not evolve? How is the genome not expressed all the time everywhere? And I think that starts, gets you thinking about selection against binding sites, but also what is the role of transcription factors or other potential mechanisms that regulate expression of gene expression levels in bacteria. And so the other question one can ask about this is, who cares? And there is some ways of caring about this, which are, to me, this is obviously very exciting and interesting and provides basic biology knowledge. But I have to say, since moving to Manchester, I'm in a faculty called Faculty of Biology, Medicine and Health. And a lot of people ask me why we care about this, and basic biology doesn't necessarily seem to satisfy them. So one way in which this is very interesting, and this is something that I'm going to use to tell you a little bit about what my group is working on recently, relates to antibiotic resistance, particularly antibiotic resistance that is achieved through the changes in the regulation of antibiotic pumps. So antibiotic resistance pumps are these big molecules that pump out antibiotics, and that can help you either give resistance by itself or help organisms transition from a non-resistant to a resistance that give them enough time to generate resistance mutations that they need to survive. And in the case of one well-studied such pump base here, AB-tol-C, it has this very complex regulation, the very complex system that regulates its expression levels. And when organisms become resistant through the changes in pumps, it's not the actual pumps that change, it's something in this regulation that changes almost always. Because these pumps are complex and to do their job, we just want more of them. And so one area that we're working on is extending the kind of constitutive models that I was telling you about to really be more predictable to more complex networks, simple regulatory networks at first, and then building to more complex networks such as this one and even further. The other thing that we are working on is actually incorporating changes in transcription factors, and not only in the promoters, that into this kind of predictive mechanistic framework. So how can we predict what the effects of transcription factor mutations might be on how promoters evolve? So this is a big project that we're working on. And here I just wanted to outline you one small application of the constitutive model that I think is really interesting, and a master student coming into the lab in a month is going to be working on this in my group, and that's focusing on, I'm going to zoom in on this one specific part, regulatory region, which is the part that regulates the actual two main genes of the pump base here, A and B. And in this system, there is a known promoter which drives the expression of this pump. And we can run our model to find and identify the model that I was telling you about before, that improved model, to help us identify all the potential RNA polymerase binding sites along this sequence. And then we can map and see whether any of those sites actually map onto experimentally identified transcriptional start sites. Right? So we try to use our model to predict transcriptional start site, and then see which of those map onto experimental RNA-seq data. And when we find, of course, there is a very notable mapping where the promoter is, so we predict the same transcriptional start sites that is also observed. But interestingly, we also predict another transcriptional start site in this kind of location towards the end of one gene and near the beginning of the other. And I can hypothesize that this can have a functional relevance. That maybe it's leading to a little bit of overproduction of ACRB because maybe functionally that's important. Maybe how it diffuses through the cell is different. I don't really know what the mechanism is, but there is something potentially happening here. And in fact, what's interesting, we find a similar pattern in many other antibiotic resistance pumps. And so we are currently exploring this by, well, we will explore this soon, hopefully by first checking whether those promoters are actually functional in an experimental setting, and also modifying these promoters to see if we see some difference in function of the pumps. Yeah, with that, I just wanted to give you a bit of sense where a few directions that my lab is moving in. I do feel a bit bad presenting a paper from 2017, but I do think I do like it because it explains the logic of it in a very simple way. Yeah, and with that, I wanted to thank everybody who worked with this at IST Austria, where I was before, and also people in my lab my lab is currently me and 4 other people, 2 postdocs and 2 PhD students, working on various aspects of this question, some on antibiotics, some on more synthetic biology questions, but all really focused on how we can predict the effects of mutations better in a variety of biological systems, hopefully. So with that, I think I did well with the time. And so with that, thanks a lot. And I'll be very keen to answer any questions.
Okay, thanks, Mato, for this talk. We already have 3 questions that seem all related to each other, in fact. So I think we're going to start perhaps with Dan Weinreich, because he's coming from the farthest. So Dan, can you turn on your microphone?
I'm going to stop sharing maybe, and then I'll reshare.
Yeah, perhaps.
Good morning.
Yes, yes. It's very early morning, I guess.
Yes, it's salutary to get up this early. I was interested at lots of wonderful things in the talk. I was interested in the very first part where you showed us the predicted epistatic effects in a simple biophysical model.
Mm-hmm.
Could you remind us why you're allowed to confine yourself to that half of the Boltzmann function, the concave down part?
The energies, so basically we, after mutating a lot of things, we could never find things that go into the, the concave and then convex, I guess. I'm really bad with these terms. So on the part of the sigmoid that would change the direction, we could never find mutants that actually had measurable expression levels there. So we could never measure expression there. There might be stuff that's happening there. The logic of it should be the same. It would just be a little more complicated. You would have to know where the inflection point is and figure out what happens around that. It's just that those energies don't seem to be relevant for what we can measure in a plate reader or flow cytometer. In the second model that I talked about, we actually we don't constrain the curve to that. We fit it on a sigmoid because there it's much more meaningful, because there we are deriving model parameters and no expression levels are also very important for us.
Okay, thanks.
Yeah, just to check, Dan Weinreich, you were at Brown before. Is this the same person or a different person?
Yes, I'm at Brown this morning.
Oh, at Brown. Sorry, I thought Louis just said Paris. That's why I was confused. Sorry.
But he said farthest.
Oh, farthest. I just said Paris. That's why no. I mean, I was a PhD student when we briefly met at the conference. That's not a slight.
Yes, I remember.
Oh, wow.
Thank you very much. Nice talk.
Yeah, thanks.
All right, so then Mathilde Delmont had what seems to be a related question. Perhaps she would need perhaps some to show the graph again. Mathilde, can you turn on your microphone and ask the question?
Yes. Hi. Maybe it's a bit a stupid question because I'm not at all used to molecular biology, but could you repeat again how the binding thermodynamics relate to mutations, like to the effect of mutations?
How the binding thermodynamics? Yeah.
So it was quite at the beginning, I think.
Yeah. So I mean, it's I'm not really showing much there, so I might as well explain it. Tell me if you want me to show the thing that I show in the background, but it's not. Yeah, I think I can do a better job in my hand. So you have a piece of DNA, and you have, in this case, let's say sigma 7 tRNA polymerase, right?
Okay.
Sigma 7 tRNA polymerase like some kind of sequences more than others, so it's attracted to some kind of sequences, and that attraction is just a product of its energy, just how the molecular forces interact. It results in some energy. For a given sequence. For any given sequence, you can calculate that energy of binding if you know the preferences of this molecule, right?
Yes.
So once you calculate that energy from, I think, so from the statistical thermodynamics theory, you can relate that energy of binding to the probability of that molecule being bound. Because if you think of, in many ways, I used to think before learning a little more about this, and I'm not a biophysicist, I should make this clear. I just learned this by working with people who are. I tend to think molecule bound, it sits there, molecule unbound, it doesn't sit there. But in reality, what's happening is you always have this process of coming on and off, and what matters is how much time or how likely you are to spend time there versus not spend time there, right?
Okay. Yeah.
And we simply say, and this is not a model, we didn't develop the statistical thermodynamic model in framework ourselves. That's something that other people, there was already a suggestion for this in the mid 80s for how to do this specifically for gene regulation. We just adapted and derived better parameters than existed before. But the idea is that the likelihood of binding, so the more likely you're to be bound, the higher your expression level. And there's really one other parameter that comes into that translation in the simplest version of the model, which is something called chemical potential, which is partly to do with the concentration of a molecule, because you can also imagine it's not just the energy of binding, it's if you have two molecules, you need less energy for one of them to be bound than if you just need one, right? So the concentration of molecule comes in there, but also a bunch of other things that are strain specific, and so that translation from the probability of binding to gene expression level has another parameter in it. But in general, is that the answer to your question?
Yes, I think it's clear now. Thanks.
All right, so now we have, I think, a question from local synthetic biologist. Guillaume, can you turn on your mic?
Yes. Can you hear me?
Yep.
Okay. So yeah, I had a general question about your data in general. So in your PR library, I guess you have a lot of relatively high expressor with moderate effect of the mutation. And in your 30N library, you might, I guess, have a lot of low expressors. So yeah, I just wanted you to comment on this and how it affects your prediction.
Yeah, so in what happens with the specific mutation rate we use for the PL library, we get something roughly even across the range. I'm trying to find, I don't want to spend too much time looking for the plot, sorry. If I can show you later or to see exactly what it is, but it's roughly even. There's like similar amount in high expression, low, intermediate, low expression, no expression. When we mutate the PR. Of course, when we have the random library, it's very heavily skewed towards non-expressing sequences, and there's very, very few high-expressing sequences, although that is still about 10% that we see in high expression bins. So it's not a super small amount. Personally, I think, and maybe this is maybe naive, but personally, to me, it's the fact that we train our model on a library where the distribution of effects is so different from the library we evaluated on, and we still get very high predictability of the random library, I think only goes to show that these mechanisms we introduce are quite powerful in explaining the variability in the data sets.
And do you know what exactly you gain? I mean, is it because you better explain the non-expressor, or you better explain the high expressor?
Yeah. So the two big factors. One is knowing the energy of changing the conformation for that different spacer length. That's a really really important descriptor, really important improvement on predictability. And because we compare it to that standard model, we still allow that spacer to change. We just don't know the energy of that change. We just say, find the best place given this feat and ignore whatever's happening in between. That's like our kind of null model, which we compare our model to where we know the energy penalty of changing that. So that's one factor. The other factor is allowing for multiple binding configurations. And that becomes really important when you have low expressing sequences. So at high expression levels, so that factor actually, so having allowing for multiple binding, or what we call cumulative binding, because we add up across all of those binding states. With the PR library, it's actually a very small contribution. So in the high expression library, that factor becomes much more important in the random library, because there's a lot more low expressing sequences. And basically what's happening is there's not any single specific site that's strong enough by itself to lead to expression, but you generate 2 or 3 of those weak sites, and together they actually give you enough binding across the 3 different transcripts. They still result in enough transcript for us to measure expression. And that's a big difference between our, the improved model and the original version.
Thanks. If I can, maybe a related question last. So you used FACS-seq to measure that, even though it's pretty remote from transcription. I mean, it's the result of transcription and translation and whatever stability of whatever.
Yeah.
So in some version of this, you could have measured transcript levels using RNA-seq and some barcodes. And I understand it's more complicated experimentally, but maybe you could actually observe like different transcription start sites for these multiple binding and things like that.
So there's a few things I can say about that. So for the paper we're trying to publish right now, this is very much what we have done and what reviewers asked us to do as well. So we used 3 or 4 different, I cannot remember enough. Sorry, 3 or 4 different data sets that were published by other people, not that we generated. RNA-seq experiments, one from just across the whole genome, some that are specifically trying to play with promoters of a specific type, and so on. And then we try to see how well our model predicts, and we find what I would think, I mean, we certainly do better than any models developed by the people who wrote their papers. For 3 out of 4 of them, much better than what they can do. Of course, that gets very complicated because you have other transcription factors binding, you have all the post-transcriptional stuff that still affects RNA levels and so on. The other thing is what a postdoc of mine is doing at the moment, and actually has spent the last 6 months doing this, is trying to map specifically to predict transcriptional start sites using our model and map them onto a range of other people's RNA-seq data. And try to see, not just to evaluate the model, but really try to tell us what we're interested in is kind of the fitness landscapes of promoters as they exist in E. coli. But to do that, we need to know which of many potential binding sites that we find are actually relevant and which by themselves lead to expression. And that again ignores what happens below the levels that we can experimentally measure, but it's a starting point that we thought was a good one. So we are thinking very much along those lines. It is also very complicated, as you say.
Yeah, thanks.
Okay, then we can move on to Tomah, who also had a question about the thermodynamic model.
Yeah, well, the question, you partially answered it, but there is one point that I'm missing here is the way you build your model, and because you said first you apply just the thermodynamics. You know the sequence, you know the energy, and so you can make a prediction of how long should the polymerase stay there. And okay, but that's from first principle, from completely external data, from thermodynamics data. But then it looks like you train your model, right? So what is it? I mean, there is not only the thermodynamics that gets into the prediction, you got something which is apparently statistically adjusted somewhere. And so it's not very clear to me you know, how you combine both the thermodynamics information and the training on the local data set.
Oh, sorry. Okay, now I understand. Yeah. So I was for a moment I didn't understand. So the training, how do we? So the training of the model. What I mean by that is actually deriving the biophysical energy parameters, right? So there is in the first study that I told you about, we actually don't even look at. We don't have a correct energy model for RNA polymerase there. We just use a kind of a dummy model, and then we just apply thermodynamics, but we don't use any, we don't know how, you know, in position 3 of -35, what is the preference of RNA polymerase given its energy? I mean, we don't model that in that first study. In the second study, we need those energy parameters. We need to know how G at position 3, what is the energy penalty of having G versus C, right? There is one study that has derived this model by Justin Keeney and Rob Phillips from Caltech in 2010. But we found that the way they, you know, the energy parameters they get we felt we could do a little better. And so that's the model fitting that I mentioned. We need to get a lot of these parameters, so to fill out what is called an energy matrix, which a more familiar version of it would be a position weight matrix, except to relates to energies in a much more real sense than phylogenetics do, like position weight matrices are made. And so we needed to derive all of those parameters. So that's the model derivation. Once we have those energies, the rest is very much just the statistical thermodynamics that I mentioned. Is this what you were asking, Tomah?
Yeah. It's clear. Thank you.
Okay, so I think we're done now. Oh no, Guillaume Cambrai had another question. Perhaps we can. Guillaume?
Okay.
One more minute.
So you mentioned at some point, just related to what you were just saying, so you're modeling as well the interactions between position in that energy model.
Yeah.
And you have enough data for that? I mean, yeah.
Yeah, we are, like, that part is definitely the weakest part of the model that we have. Let me put it that way. It's most ad hoc. We had to make a lot of assumptions there. And personally, if you want my opinion, if you use the model that we have, I would kind of not trust that part as much.
Okay, because that's very much related to epistasis that you talk about, right? I mean, it's on a different level, but
yeah, I mean, what I'm personally really interested in is sort of mechanistic origins of different types of epistasis. So one is just that kind of what I think in my own head is stupid epistasis. You know, epistasis that just comes because we define something based on some fictional mathematics that has nothing to do with biology. There has to be some epistasis at the level of energy. Exactly how that works would be interesting. And just ranking those different mechanisms that contribute to epistasis is a pet project of mine that I'm trying to persuade somebody to work on, but I'm getting too many medically minded students lately. So.
All right, thanks.
All right, I think we'll leave it there. So thanks again, Mato, for this nice talk. And too bad you couldn't come, but I think this will be postponed to another occasion. So I just want to let everyone know that next week we'll have something a bit special, because it's Montpellier Global Days. And so Muse and the LabX is very much involved in that. And so we'll have a SEM that will be later in the afternoon, for exceptionally at 3:30 p.m. And then we'll have 3 speakers who are doing research in ecology and evolution in Africa. So this will be an interesting, make sure to join. And with that, I'll see you next week. Bye-bye.
Thank you so much.